Oncology. Custom tailoring cancer care from patient to patient is an exciting and potentially revolutionary path forward for both patients and cancer care centers. Here to expand on this and lead this discussion is Dr. Mauro Ferrari, a noted nanoscientist and active entrepreneur. He has served as a tenured professor in engineering and medicine at the University of Texas, University of California, Berkeley, The Ohio State University, and Weill Cornell Medicine. For nearly 10 years, he was president and CEO of the Houston Methodist Research Institute and served as executive vice president of Houston Methodist Hospital. Most recently, he presided over the European Research Council. Dr. Ferrari's own work is in nanomedicine, transport, oncophysics, and nanotechnology. His laboratory is actively engaged on these fronts, in particular, an endeavor to develop cures against metastatic breast cancer. Please welcome our moderator, Dr. Mauro Ferrari, and he will introduce our panel.
Well, good morning, everybody. What a delight and privilege to be here. I'll give you a couple of seconds to get used to my Italian accent. And I'm going to be copying Dr. Pister with this idea of introducing everybody, letting everybody introduce themselves with a little fun fact as well. I think that'd be a wonderful thing to get the conversation going. So we are focusing for this panel on the concept of precision oncology. And of course, precision oncology is a word that came into existence perhaps some 20 years ago. Of course, with the promise that it would be a transformational change in the fight against cancer, and in many ways it has been. Some of the early expectations, of course, became reality with new drugs that were molecularly targeted coming into clinical reality. And I'm thinking of Gleevec in 2002, I'm thinking of Herceptin, and so many other of the drugs that you're all very familiar with. Now I think is a good moment to reflect about how far we have gone with respect to the expectations that we had perhaps 20 years ago, recognizing all of the successes, of which there were many, and taking inspirations from the things that have fallen short, so that we can reload and update our strategy and move forward towards serving patients in the very best possible way that we can. Stealing from Dr. Pister's approach, I'm going to start with a fun fact myself. And as some of you that were here last year, this is my third year here, it's a great privilege, it's a great honor. But as some of you may remember, if you were here last year, I sing, all right. I know it's a painful memory for some of you. Yes, but I do sing, and I keep on singing. I've been doing this. And I sing about science. I sing songs that help me reinforce the notion of the scientific communication that I try to do in the many venues where I speak. Because the reality is that intellectually we may be connected, information may be flowing both ways, but to really stick, we need the emotional connection. The heart always wins over the brains in so many different ways. I'm Italian, so I can say that, right? And so I do, many times I do these shows where I talk to big audiences together with an orchestra, and we sing and we talk and we show slides like we would do at a scientific meeting. And I found that that's a great way to engage the community. But not because I sing well. You remember that. Those of you that were here last year remember it very well. It's because I don't. Exactly that. Because somehow, seems like people trust more a scientist that is willing to show his or her limits and to have a good laugh altogether. So there is nothing to laugh about the topic that we're going to be addressing, of course, today. But I think some of that notion of managing and moderating expectations and recognizing that the trust that we can build with the community is based on the truthfulness that we show in terms of successes, but also failures. That is what I would like to bring to the conversation today. And with that in mind, it is my great pleasure and delight to let my panelists introduce themselves. And as you already know, this is a spectacular trio of very accomplished scientists and clinicians. Christina, why don't you take first?
Sure. Hi, everyone. I'm Christina Annunziata. I am currently Senior Vice President of Extramural Discovery Science at the American Cancer Society. Before that, I just started 6 months ago there. Before that, I spent 21 years as a clinical researcher and laboratory researcher at the National Cancer Institute. So I have experience both in clinical research, laboratory research, and now funding research. So my fun fact, we didn't have much time to prepare for this. So my fun fact,
that's what makes it fun,
I guess, is that, so my favorite type of book to read, my literature genre is sci-fi fantasy. So my favorite book would be someone with magical powers who rides a dragon and fights with a sword. So that's my genre.
I'm tempted to ask you about your favorite music, but we'll do it some other time. You know, Christina is sensational because, of course, not only she did the science, but she was able to bring it into clinical fruition, which is an incredibly complicated task. I've been engaged in contributing to the best I can to doing this, which we call translation. And my personal experience, now that I also work on the industry's pharmaceutical side, my personal experience is that sometimes translating your discoveries into the clinic is a lot harder than doing the science itself. It certainly takes a lot longer. It certainly takes a lot more money and a lot of stick-to-itness and refuse to die type of attitude for the many hurdles. But we need to be able to conquer those if we want to serve our patients, which is what we are here to do. And she's done it in the context of ovarian cancer in multiple different dimensions with stem cells and the NF-kappa-Bs and the SMAC inhibitors and mimetics, and with intraperitoneal, of course, immunotherapy. So that works, and I really look forward to learning from you in this panel. Sourat?
Thank you. My name is Sourat Darabi. I'm a clinical genomics scientist working as a director of precision medicine at Hoag Family Cancer Institute in Orange County, and I'm honored to be here with all distinguished researchers and scientists. I'm also running the Translational Laboratory and Early Detection programs at Hoag. Fun fact about me, again, that's fun, but it's not probably about me, but I have 2 teenagers, and I'm learning a new language. I went from mom to bro, and I'm learning Riz. Anybody know what Riz is? Okay. So in 2023, that was word of the year. Means charisma. So this is a new language I'm learning currently, other than what other stuff that I do. Thank you.
Fantastic, thank you.
I'm Linda
Please,
Malkas.
Linda.
I'm the Dean of Translational Science at the City of Hope. I am one of those people who discovered or noticed a funny band on a gel, which turned out to be an undruggable protein, which I then drugged and is now in phase 1 clinic. So, yay. Fun fact. So, fun fact, I'm going to borrow a little from Tina here. I am a massive Star Trek fan. I do have a uniform, and I have gone to the conventions. But the other thing is I'm also responsible for 180 horses. We can talk about that later. Okay.
Fantastic. Thank you so much, Linda. So, again, the word precision oncology emerged about 20 years ago, and it caught on like wildfire, riding on some of the successes and riding on the fact that nobody wants to be the champion of imprecise oncology, right? So, of course, then everybody says, look, I want myself some of that. I'm a champion of precision oncology, of course, is the right thing to do. The right medicine at the right person at the right time. But let's look at what foundations the concept of the right medicine is based upon. Initially, it was all of the omics. And then perhaps it expands rightfully or wrongfully. I don't know. I'm not here to judge. But that is why I would like to ask start asking our panel with what is their definition of precision oncology. Would you like to start?
Sure, I will start. So I think when I think of precision oncology, there's three main aspects. One is the cancer itself, second is the drug, and the third is actually the patient. So what about the cancer itself? Of course, we have the omics. We need to know what mutations are there. We need to know what different proteins are modulated, what networks are disturbed. So we need to understand the cancer. We also need to know the difference between the primary cancer and the recurrent cancer, the evolution of the mutations that occur and the changes that occur with various treatments, the treatment history, and how the cancer has changed. So we definitely need to know a lot about the cancer in order to be precise. We also need to know about the drug. We need to know how the drug is administered, metabolized, how it interacts with other drugs, what order it should be given if it's combined with other medications, and how that affects its metabolism and its efficacy and its delivery and its delivery into the cancer itself. And then the third aspect is the patient. So the patient, not just the patient's genetic makeup, but also the patient's behavioral makeup. What foods do they eat? What is their diet like? What is their lifestyle? What are their other comorbid conditions? Are they taking other medications? And how does this drug regimen fit into that patient's lifestyle? So I think all of these three things come into my definition of precision oncology.
Thank you very much. Sourat?
Thank you. I second what Tina said here, but you know, every one of us here in this room, if you ask them for a definition for precision medicine, that we might have one common, is a model to better help patients. And that can be from early detection. Early detection is also important in precision medicine in general, and here we talk about oncology, precision oncology. Early detection, how we can treat the patients, monitor the treatment, and also monitor recurrence.
Very good.
So, precision medicine, I look at it, and it really has been a revolution in the last 20 years. I mean, we have so many examples of really astounding, beautiful targets where we develop therapeutics to it. But you know, just like the last panel, we're talking about AI, and it just seems so huge. For me, precision medicine is just as huge because you're looking at systems. You know, I live inside the cell nucleus, and so and then I venture out, you know, into the cytoplasm and stuff like that. But touching on what Tina talked about, you know, you have, you know, I found a target. But when you start to think about, you know, this target is modified by something called post-translational modification, because everything is resting on genomics. But then, yes, we have mutations and targets that way. But there is an entire sea of proteins that are post-translationally modified, which we have really barely ventured into. And then actually the thoughts of how, you know, you have the tumor. You know, we always think tumor, non-malignant, tumor, non-malignant. But there's also this entire environment around a tumor that is communicating. It's system on top of system on top of system. And I still feel like we're like in a baby place yet for really beginning to understand where and what precision medicine is and where it can go. That's just my thoughts.
Thank you so much. Now, let me start to challenge a little bit our respective notions of precision medicine and see if all together we can envision what it takes to continue to do better, which is always our challenge. So as I'm looking at the beautiful charts that the ACS puts out every year about mortality rates, since the 1920s or something like that, I look at them, and you all remember them, you all seen them many, many times. There are two big dips, two spectacular dips that are associated. They are divided by diseases, right? They are associated with two objects they were very familiar with. One is the refrigerator, and one is the cigarette, right? changes in the way society behaves. In the case of the refrigerator, simply because you have access to fresh foods, so much healthier. And the second, the cigarette for cessation programs that have been championed by the government and many other entities. Great, those are the two huge dips. Fortunately, because of the work of so many people in so many different areas, we have also seen a steady decline in mortality pretty much across the spectrum, with the exception of pancreatic and perhaps some rare tumors. And that, of course, that decrease in mortalities is more or less where we sit with precision oncology as well. It is one of the components. I haven't seen with the onset of precision oncology a big dip all of a sudden, which is what we all have in our collective imagination as a community, that at a certain point, some very smart scientist or lucky scientist is going to come up with something, boom, where you see that mortality drop suddenly. It doesn't happen that way. So we need to see ourselves as precision oncology, I think, champions and advocates in that context of the incremental, of course, progress. So my question for you would be at this point, what do you see within this context, or outside if you prefer, as the big successes and the big failures? And I'm saying failures as, of course, junctions along the road where we have the opportunity to take perhaps an even more potent road, and not in a critical fashion, but in a constructive fashion. Why don't we reverse? Would you like to start, Linda?
Oh, of course. Okay, so, everybody's waiting for the magic bullet. At least that's what I grew up with, right? You know, and when I was trained in the last century, the last millennium, it was, you know, we're going to find the oncogene. Well, there's lots of those. And then there was something on top of that called tumor suppressors. And then everybody was looking for the magic bullet. And in my opinion, there will never be a magic bullet. We're going to have lots of bullets. I hope I created a bullet. I'm hoping it was a rocket launcher myself. But I think and we touched on, some of this was touched on in the talk last night, but I think where the magic is going to happen is when we just start to do really great combination therapy. There's still a lot of weight on monotherapy. You know, like they're going to just do it with monotherapy, and I don't believe, especially with a lot of the targeted therapies that we're coming up with, that we're just going to rest on one drug for everything. You know, there's a great energy around certain therapeutic approaches, but it's going to be the dance and, again, the patient individualization that you were talking about. That's just kind of where I'm thinking it.
Thank you. I will come back to combination therapy later, perhaps. Sourat?
So this is, you know, as I was listening to the previous panel, and this is what keeps me up all night. We have all these great tests. We do great as much as we can to care for these patients. We don't have a good tool to bring all this combination together, and that's what AI, I'm hoping, at some point is going to help us. Now we do multi-omics, we do proteomics, we do, germline genetic testing, we do somatic testing, we do sequential testing, and we have all this series of imaging. These are all precision medicine, but then put them together, how we can come up with an idea, okay, what is going to help this specific patient? As it was mentioned, a lot of them are linear. We don't know how we can put them all together and come up with, okay, this drug might have a better response to this patient versus the other one. I think what we're lacking is a tool that help us to put all these information that is valuable that we so far have together to come up with a better course of treatment and monitoring for our patients. I think AI is something that I'm really looking forward to help precision medicine.
Allow me to do a quick follow-up with you, if I may. Now, you brought up early detection in the context, of course, of precision oncology a couple of times already. Can you expand on that a little bit? And what do you see is missing? What are the directions for growth that we should be all taking up?
Sure. So we all talk about patient as a whole, and we're trying to help them. We do all this research. We spend billions of dollars on trying to treat patients. But what if we can detect it early? We talk about pancreatic cancer. We normally see them in a later stage. How could we have early detection with if we have an early detection algorithm? That's what I call it, algorithm, not one single blood test, not only genetic testing, not putting family history together. We want to have an algorithm that put all the information together and tell us this patient needs to be monitored this way. And then we can detect pancreatic cancer early. So we can do the surgery and have a better 5-year overall survival or 10-year survival for these patients, comparing to when we actually detect the cancer is too late. We're trying to, it's not cure, but we're trying. So early detection in terms of germline genetics, environmental factor, where the patient lived, where they born, and how they raised, and what they were exposed to, I think is also important to put all together for early detection. I think focus on early detection. I'm not saying that we need to move away from treatment, but if we focus more in the next decade or so on early detection, we're going to have a better precision medicine in terms of oncology, precision oncology.
Thank you. Tina, successes and failures and directions.
Yeah, so I think, we're getting, we're sort of blending precision oncology with personalized medicine, but I think that's where we need to go, is personalize the treatments. I think certainly success, as you mentioned, there hasn't been huge drops in mortality across the board, but there have been huge drops, huge successes in individual cancers that might be rare, but we really have hit the target on those, such as CML with Gleevec, you mentioned that one. And then some of the other newer target ages. We're targeting a very, very small population with great success. So I think that's why we're not seeing, like I said, like a huge overall drop in cancer mortality, but individual diseases, molecularly defined, have had great successes. I think where our shortcomings are, if we could get recommended treatment to every patient at the correct dose, I think that would be a huge success in making a big impact in mortality. So for example, even with radiation therapy, people can't get through their radiation therapy for one reason or another. Radiation therapy is standard upfront treatment for cervical cancer, for endometrial cancer, for several other cancers. But a lot of times people can't get to their appointments. They can't tolerate the radiation. They can't tolerate the chemotherapy that's given in conjunction with the radiation. So how can we better get people the standard treatment, the FDA-approved treatments? I think that would be more of a personalized approach to medicine to get those people through. Do we need more patient navigators? Do we need more chat bots? Do we need more robots in people's homes to get them through? These are ways that maybe we could make a more personalized approach to getting patients their standard of care and having a big impact on mortality.
Thank you so much. Now, if I can add a little bit to the conversation, there is a couple of aspects where we're talking about translation. And I had a big wake-up call on translation, even though that's what I've been focusing on pretty much my whole professional life. I've had a big wake-up call again on the timelines. Last week, I was at JP Morgan, and I saw every single one of the CEOs and major leaders of every single major pharmaceutical company worldwide that deals with oncology. And the recognition came that as they were presenting and they were talking about the strategies going forward and the history of how they got to where they got, many times it was research programs that had been going on for 15, 20 years. So perhaps one reason why we haven't seen the drop yet is that it takes from the onset of the scientific recognition to the moment of clinical reality, it takes 15, 20 years and more. So we have seen just a little bit of the science that's been generated since, just a little tiny bit, which begs the question, what should we do to render our clinical trial machinery, of course the discovery, but then development and then deployment? What can we do? And of course, Dr. von Eschenbach has been a legendary champion of that. What can we do as a community to facilitate an equally accurate, or perhaps even better, translation of scientific discoveries into clinical reality? But the second point I would like to bring up, a bit of a provocation for you, of course, great scientists, and for anybody that wants to pick it up. Later on, we're going to have a little Q&A, is that here we are looking at our understanding of cancer at the gene level. Great. That's where it all started. That's a necessary component. And of course, targeting defective genes after we have done our genomic screens, and doing associations, and doing correlations, and all of that is very important. Kind of makes me wonder, though, to take the next step up, and this also ties in with the combination therapy approach. Are we really looking at cancer as a disease of genes? Or are we looking at cancer as a systemic disease, of which, of course, genetic alterations are a fundamental component, but driven perhaps inside of a greater notion of balance that can be tissue level or can be systemic level, having to do with phenotypic expressions that are perhaps more connected to the true reality of what these many diseases are that we call cancer, than certainly than the traditional division in terms of site of origins, right? And I'm thinking maybe it's metabolism, maybe it's imbalances in transport, perhaps genes. I mean, great if we find this gene is important and that gene is important. Personally, I'm curious about finding a gene that is not important at all in cancer. And why? You know, because it kind of points to the need for greater complexity, if you will, into the understanding of what the key fundamental disease processes and their drivers are. And how do we get there? One thing that I would like to throw in terms of precision medicine, of course I'm biased, is mathematics. Is the physical approach of starting with a foundational principle and then looking at these derivatives, which allows you to postulate hypotheses and then to verify or falsify them doing experiments, as opposed to going on a search that comes from a screen, which is very helpful, but is a bottom-up that does not take into account necessarily a top-down perspective. And the other thing that I think is necessary for this, which is the topic that I was hoping would come up in artificial intelligence, and that's where it is. That's where it is. You don't need to build mathematical model from the bottom. You can use applied statistics to find some of the general, of course, correlations that then can give you an idea what is the basic fundamental master equation of cancer. And my impression is that that would not be at the gene level, it's at the tissue level, but probably so at the genetic level. So that was just my personal, Linda.
Well, you know, actually, I like what you're saying here. Again, it goes back to kind of like what I was trying to, but you did it much more eloquently than I did, which I was talking about is a massive systems problem. You know, we make cancer cells every day. We're all cancer survivors in a way. And so like you're saying, you know, it's a balance. We fend it off every day. But when, and then here's your, this is early detection, but how do you do that? You know, what tips it over so that, you know, you go from having this steady state of, you know, your immune system taking care of you to where you know, I don't know if we really have done a lot of work in that area. You might know more about that. I would appreciate hearing, you know, what tips the balance. Sometimes I have thought some of the people we should be studying are these magnificent beasts that actually have fought off like 4 different cancers successfully. It's kind of like we should be studying them, you know, because they may hold a lot of answers with with this systems balance kind of thing that you're talking about.
And the gene mutations almost can be seen as reactions to the offset of the balance. Is that what you're thinking? And where that connects, of course, with the notion of multi-drug cocktails, as you were talking about, combination therapies, is that if you look at how those magnificent beasts, humans and animals alike, are more successful at finding cancer, it is never with a single molecule, right? It is never as a combination of molecules and cells. And other dynamics that we probably should be looking into a little bit more in terms of, again, balances. Thank you so much.
So, great points. So when we talk about genetics and genomics, I just want to make sure we're not talking about just mutations. We're talking about genetics, epigenetic effect. We are, as you mentioned, we are in infancy here. It's a decade of work, but we still really don't know what to do when, especially I review thousands of reports for somatic testing and also germline a year. I see resistant mutations show up, amplification, fusions. What I'm gonna do with all the combination, there are few studies showing, okay, if you have an EGFR T790M, you need to treat with this drug or any other ones. But really, we don't know. And as you mentioned, we need to talk about immunology, inflammation, something that is beyond what we see as a mutation or any alterations in a gene, but it's, what it affects, like, is not oncogene or tumor suppressor gene, but it will affect the proteins that about inflammation and how the immune system responds. I think this is really important. Like, when we go back to germline genetic testing for hereditary cancer, over a decade ago, we just did BRCA1 or 2 mutation testing. Now we have panels. We do exome sequencing. We do genome sequencing. We have all this data that we're looking at. So there's a new studies looking actually not the mutation. Some patients with BRCA1 methylation, they have hereditary cancer. So we're learning as we go. So I think when we think about it as a big picture, as it was mentioned by the panel, I think it would help us, but I think we're way to go to get to the point that we really want to be. We need to study more and understand everything together, not separately.
Yes, I mean, definitely building on that systemic, I completely agree with you. We are learning. We're learning as we go, and definitely I echo the other two panelists about the systemic nature of cancer. It is a tissue disease. It's not an individual gene. But if you think about the immunotherapy, I mean, we have a lot of cancer immunotherapy out there right now, whether it's immune checkpoint inhibitors, CAR T cells, or other sort of off-the-shelf cellular immunotherapies. Vaccines are out there that are FDA approved. These are systemic treatments that are targeting the phenotype and not the genotype. They're targeting either a normal protein that's overexpressed or a mutated protein that's expressed by the cancer. They're not targeting the gene itself. The gene might be the responsible thing for getting that protein on the surface of the cell, but the immunotherapy is really working via the abnormal proteins or the normal proteins that are expressed on the outside of the cell. So if you think of immunotherapy, it can be considered a targeted therapy, a precision medicine, because it is targeting a specific mutation or protein that's expressed on the cancer cell, and it's using the whole body to fight that away. The way perhaps other cancer survivors, that survive cancers, low-level, undetectable cancers, are doing it all the time. So the way our body takes care of it is the immune therapy, and that's what we're trying to manipulate on a cancer-specific level.
Fantastic. So I think through the conversation, we have included in the notion of precision oncology components of personalized oncology, of course. We are looking at including at least components of immunotherapy, of early detection and the technology that goes into that. I propose math and physics of transport. Anything else that we have missed that should become important and prominent in precision oncology going forward?
I think the next panel might touch on that.
The next panel will touch on that for sure. That's why I talked about the refrigerator first. I want to be friends with them, yes. Anything else you would like to add?
Just that, I think we can't talk about precision oncology without talking about diversity in clinical trials. So I think the other panel also brought this up. We need to understand differences between individuals and not just sort of assume everyone is the same. So I think the FDA has a new initiative for diversity in clinical trials. It's a starting point, but it's certainly some knowledge that we need input in order to be able to process that and understand how better to treat cancer on an individualized basis.
Outstanding. I'm looking forward to coming back to that in a moment. Anything you would like to add?
I think also something that in terms of not looking at science is availability. I think what we're lacking currently is what we see in some rural areas that availability I think is important to also consider.
I think access is as fundamental, if not more fundamental, than anything else we have talked about on the science side, because that is where the ethical responsibility for us all as a community resides. So I certainly agree with that. Of course, we can also mention things like gene editing. We can look at the gene therapy of the RNA type through vaccines or through silencing approaches. There are many new tools that I trust will continue to be featured and will become more and more prominent in this broader umbrella, if you will, of precision oncology. But let me ask you really quick. You have all been working intensively on the translational side. So can you share with us your personal frustrations, if any, and use that as a foundation to propose things that could be done better to move discovery safely and effectively to the clinic in a more efficient fashion? Any frustrations, or am I the only one that has experienced frustrations in translation, of which I have many stories to tell?
I think the frustrations in the clinical arena, in translating to the clinic, is that you have a set of very distinct findings in the lab, and then you have a very diverse population that you're trying to administer that to. Whether it's, when you're doing your mouse studies, they're all female mice, and then you take that to a male and female population. It might not work out the same way. I think that's probably the biggest frustration. Other frustrations would be getting access to the experimental drugs that you want to use in the clinical trial. So that's a whole other regulatory aspect that can be very frustrating. But I'll turn it over to the other panelists.
No, I agree what Tina said, but I think we all, other than also funding, is another, could be a limited factor for some translational research. As Tina mentioned, dealing with pharma companies and mouse models and bringing the everything, it costs these days so much. I think if it was not for cost, I think we would do much better work in a translation or oncology research than what we do right now.
I read in some place that the two rarest things to find on the face of the earth, one is a gene that is not involved in cancer, and two is a scientist that doesn't think that more money should come to science. I think we have talked about both. Linda?
Well, actually,
I agree.
I've had the great privilege of working with a wonderful medical oncologist named Dan Von Hoff. And he's been a phenomenal mentor to me. And so when I got the approval, I got the email that the FDA had approved our doing the clinical trial. Dan was so sweet. He said, Linda, I'm going to send you something right now. I want you to look at it. And I go, so he sends a file, and I open it up, and he's on the phone, and it's a Gantt chart. You know what a Gantt chart is? It's like this chart. And he goes, so it's 6 pages, this Gantt chart. And it starts with naming the compound, and it ended with dosing the first patient. And there were 350 steps. And he says, Linda, you're at 349. And he goes, and I said, Dan, I'm so glad you never showed this to me before, because I don't think we would have got here. And that is part of the problem.
Of course. Thank you so much. I've been advised that we only have 5 minutes. and I certainly don't want to deprive you of the opportunity of asking questions.
Thank you. Thank you to the panelists. It was a really good discussion and the moderator. So I have two questions, one to the panelists and one to the moderator. If the panelists can comment on precision medicine in the realm of the tumor microenvironment, what's being done to address in a very precise way things like tumor-associated macrophages, T regulatory cells, and the like. I think that's an important aspect and maybe something we can discuss. And to the moderator, given your expertise in nanotechnology, nanomedicine, and drug delivery specifically, how can we use those tools to augment the delivery, more effective delivery of precision medicines?
Thank you very much. Anybody would like to take the first question?
Okay, sure. Yeah. So the tissue microenvironment is extremely important variable that is not is often overlooked. You mentioned the immune cells. Certainly, we need to get rid of the immune suppressive cells. We need to bolster the trafficking of the CAR therapy, if you're using that, the trafficking of the T cell into the environment. Other things that we might not think about is the acidity of the microenvironment. It could be that the tumor is very hypoxic, and then the delivery of the cells, the drug, the protein, is going to degrade and not get to the tumor microenvironment in an effective way. I mean, these are all various things, the blood vessels that are going into the microenvironment. We talked about angiogenesis, I think, last night. What's going on there? How can we optimize that? I mean, I think all of these aspects are being studied, but I think our challenge is to bring them all together and try to figure out what needs to be done at the exact moment.
Yeah, so this is a great point. Tumor microenvironment is very important and critical. Unfortunately, not a lot of clinical testing is available for that. I really would like to see that I see some of my colleagues from different laboratories here and encouraging them to put that on a report, because what I want to see, in an ideal world, I want to see when the patient's diagnosed with cancer, I want to see full profiling from tumor microenvironment, what is clonal, what is subclonal, and then see the evolution throughout the disease progression or treatment. So tumor microenvironment, other than the clinical research setting, not much available in a clinical setting. But I agree with you, we need to have those available to us for patients. Like if the cost was not an issue and we had the availability of all these tests, we would do all of them up front. So that's one of the topics we didn't get to talk about, is how we can improve precision medicine in terms of, local private hospitals. We all talk about clinical trials, and all can be depending on tumor microenvironment, clonal, subclonal aspect of the tumor, as well as the big picture of the profiling.
Thank you. Be very happy to give a super quick answer. Yes, nano certainly is a component of personalizing, and we have dozens of new drugs that have come ever since Dr. von Eschenbach launched the Cancer Nanotechnology Initiative, and without which, by the way, I don't know that the mRNA vaccines would have come into existence as quickly as they did. So the far-reaching implications of what the gentleman did, I think, are many. But certainly, nano is now become so diffuse and so broadly distributed that they really get subsumed under the notion of formulation, which we do for all drugs, for any drugs. So it is present in so many different ways that nobody even knows about. And I think that gives me good hope that it can continue to develop into something more important. They don't do nano stuff anymore. There are many things that you can do after that, but they don't refer to the notion of transport, of mass transport in the body, which I think, if there is a master equation of cancer anywhere, I would be surprised if it did not involve the transport. And that is why I think these type of strategies are going to become more and more important for sure. Do we have time for another question? Okay, good. Okay, I'm Italian. I keep on going. I'm sorry. So I thank you all very much, these great panelists of ours, and I thank you all for your attention.
Thank you to the panel. We're going to go take a quick break, and we will reconvene in here at noon with the UCI luncheon and our keynote speakers, Joan London and Julie Gerberding.
Concentration was, can you think
Can I get a quick picture with all of you?
Of course.
Good isn't good enough. It's your health.